My name is Betsy Locken, and I am an undergraduate biology student at the University of Alaska Fairbanks. Today I will be presenting on the implications of the Bult-Ito OCD mouse model. So OCD is a neuropsychiatric heterogeneous disorder, meaning that it can differ significantly from individual to individual, and a mouse model would greatly help to understand the neuronal pathways in OCD and also help to develop new treatment options. So our purpose of this study is to test the validity of the Bult-Ito mouse model to show that it is able to give consistent results, and specifically we are going to test the compulsive-like and anxiety-like phenotypes of the mice in the model. So OCD is the fourth most prevalent mental health disorder in the United States, affecting approximately 1 to 3% of the population. It is characterized by ongoing obsessive thoughts and compulsive repetitive behaviors. So obsessions include things like fear of contamination, need for order, and pathological doubt. And compulsions are behaviors like hoarding, washing, seeking, counting, sorting, and checking. So the causes of OCD are not fully understood, and they can be unique to each individual, but it is shown that OCD has a 27 to 47% heritability rate, and studies have shown that there is a pattern of a disruption of the corticostriato-thalamocortical circuit, and they've also shown that there can be hyperactivation of the orbitofrontal subcortical pathway that leads to exaggerated concerns about hygiene and danger. And these exaggerated obsessive concerns can lead to the compulsion-like behaviors and also clearly would cause a lot of anxiety in a person. So some of the comorbidities that people with OCD often also have are anxiety, depression, panic disorder, bipolar disorder, attention deficit hyperactivity disorder, body dysmorphia disorder, hypochondriasis, Tourette syndrome, Parkinson's disease, catatonia, and autism, with the most prevalent of these being anxiety, at a rate of 75.8% of people with OCD also having anxiety. So the treatment options available include therapies and medications. The most common therapy is cognitive behavioral therapy, and some of the types of medications include selective serotonin reuptake inhibitors like fluoxetine, tricyclic antidepressants like clomipramine, antipsychotics like risperidone. And even though there's all these treatment methods available, it is still shown that 40 to 60% of people with OCD are actually resistant to the available treatment methods. So the Bult-Ito mouse model is composed of 3 different behavioral strains of mice, and each of these behavioral strains has a replicate, so for a total of 6 mouse strains. The HA strains are composed of mice that were selected to build large nests, and this represents a compulsive-like behavior. The LA strains are composed of mice that were selected to build small nests, and this represents a non-compulsive-like behavior. And the CA strains are a control strain of mice that were randomly bred, and it can be compared to the other strains. So it is interesting to note that the original study done by Bult and Link in 2000 found that the big nest builders used approximately 40 times the amount of cotton than the small nest builders, and only male mice are used because they show higher levels of OCD-like behavior than the female mice, and they were used at around 50 days of age. So to assess the Bult-Ito mouse model, we used 4 behavioral tests. 2 of these tests assess the compulsive-like behaviors, and 2 of the tests assess the anxiety-like behaviors. So for the nest building test, which is assessing the compulsive-like behavior, we did this by putting the mice in a cage for a 24-hour period with a ball of cotton on top. And the mice were able to grab at the cotton and build nests, and we assessed how big their nests were by weighing the cotton before and after the 24-hour period. So the amount of cotton that they used was measured in grams, and this was their nest building score. So here you can see a picture of their apparatus, and this is actually from one of the HA compulsive-like strains of mice, and you can see that they built a very large nest. So for the marble bearing test, we did this by filling a rat cage up with wood shavings, and then we placed 20 marbles evenly dispersed on top of the wood shavings. The mouse was then placed into this cage for 20 minutes, and we recorded the number of marbles buried at 5, 10, 15, and the 20-minute time mark. And marbles were counted buried if they were at least 2/3 of the way covered by the wood shavings. So this test assesses compulsive-like behavior, specifically compulsive-like digging behavior in the mice. For the open field test, it is measuring anxiety or fear-like behavior. So mice were placed in the open field for a total of 3 minutes, and we assessed 3 different behaviors. So at the beginning of the test, the mice were placed into the central square, and here you can see this is the central square of the open field arena, and it is composed of these 16 squares that are 10 by 10 centimeters each, and the central square is composed of the inner 4 squares. So the first behavior that we measured was latency to leave this central square. So this was measured as the time that all 4 paws had crossed that line and left the center square. We then measured the time spent in the center square after initially leaving the center square. And then we also recorded locomotor activity, and we counted this as the number of lines crossed during the entirety of the 3-minute test. And we counted a line crossed if all 4 paws had crossed the line. So here is the elevated zero maze test, and you can see that it is elevated 50 centimeters above the ground, and there are 15 centimeter high walls on either side. So the mouse was placed into this enclosed part of the maze initially, and we counted the anxiety-like behavior as the time that the mouse would spend on these open arms here and here of the maze. So as soon as all 4 paws had left this enclosed quadrant of the maze, we started the time, and we only recorded the time that the mouse was actively exploring this part of the maze. And you can tell there's cotton down here on the ground, and that was just in case any of the mice fell off. And it is important to note that some of the mice did fall off the maze, and so they were excluded from our data points. So before the test took place, we measured the body weight of the mice for each strain, and as you can see that the HA compulsive-like mice had the highest body weight, and the LA had the lowest body weight, the non-compulsive-like mice, with the control strain having an intermediate body weight. For the nest building test, our results showed that the HA strains built significantly the largest nest compared with the CA and LA strains. And you can see that the LA, non-compulsive-like mice, built the smallest nest, with the control being in between. For the marble bearing test, you can see that the HA strain, the compulsive-like mice, buried the greatest amount of marbles, and the control mice had the next greatest amount of marbles, and then the non-compulsive-like mice buried the least amount of marbles. You can also see from this graph that as time went on, the number of marbles that got buried increased. So for the open field test, here are the results for the latency to leave the central square. The results were not significant, but you can see that there is a general trend here. The non-compulsive-like mice actually took the longest amount of time to leave the central square. So this shows that the non-compulsive-like mice are showing the highest levels of anxiety for this test. And for the total time spent in central square, our results were not significant, although we did have some replicate effects showing differences between the replicates, just giving you an idea of how important it is to replicate your data, and it also signifies when we did have findings that they are even more backed up by our replicates. And for the number of line crossings or locomotor activity, there also was no significance between the strains. And we did have another replicate effect here between the LA strains. For the elevated zero maze test results, you can see that there was a significant effect for the strains, the HA strains. They actually spent the most amount of time on the open arms, showing that these compulsive-like mice actually showed the lowest levels of anxiety. So, between the nest building and marble bearing tests, we were able to confirm our hypothesis that these compulsive-like mice would show the most compulsive-like behaviors. So this is good and shows the validity of the Bult-Ito mouse model. It was also interesting that we found that these mice had the largest body weight. There's different hypotheses that could also be discussed, like the fact that maybe these behaviors are behaviors that would be consistent with animals who are living in colder climates. They may build larger nests, they may dig more to keep warm, and also have a bigger body weight. So maybe these cold-like adaptations for colder environments are also linked to the compulsive-like phenotype. So for the anxiety-like behavior, for the open field test, there was not significance, but we did find significance in the zero maze test, and it was interesting to compare both of them. Even with the open field where there wasn't significance, we still found the same general trend that the zero maze test found, and that was that the non-compulsive-like mice actually showed the highest levels of anxiety. So this goes against what we would have predicted, and this is showing that that for the Bult-Ito mouse model, the mice that have the highest levels of compulsive-like behavior have the lowest levels of anxiety, and the mice that have low levels of anxiety or have low levels of compulsive-like behavior actually have the highest levels of anxiety. So this is interesting to think about, and it could suggest that the Bult-Ito mouse model may only be good for people who only have OCD or only assessing these compulsive-like behaviors of OCD and not the comorbidity of anxiety. Or it could lead us to think perhaps that maybe there are differences involved in the anxiety that comes along with humans with OCD and with mice. For instance, humans know that they have OCD and they know that their repetitive behaviors are abnormal, and this could cause them to feel very anxious and a lot of anxiety, unlike the mice who don't realize that there's anything wrong. So just some interesting things to think about when looking into our results. But overall, we were able to confirm that, to show that the Bult-Ito mouse model had consistent results throughout all of the tests, and we were able to confirm the compulsivity-like behaviors in the compulsive mice. So I'd like to thank Bult-Ito especially for the contribution to this